When choosing a movie you're going to watch online, you're going to have a plethora of choices. You can, of course, just type a name into the search box to find something specific. However, if you don't really know what you're going to watch, you can choose a movie based on the category it is in. There are horror movies, dramas, comedies, and so on. In the same sense, viruses can be classified or categorized based on certain features they may have. Some are dramatic, some are horrible, and all are out to get you, sort of. In a very basic sense, viruses are classified based on their physical and chemical characteristics. Unfortunately, that's just the basics. There's way more to it than that, and we'll only cover the important points of what is known as the Baltimore classification system, which classifies viruses based on their type of genome and method of replication. For instance, viruses may have a nucleic acid genome that is either DNA or RNA, either of which can be double-stranded. Double-stranded viruses are those that have a genome that consists of two chains of complementary nucleotides that are joined together. I know that was a mouthful, so let's break this down into a simple example. DNA or RNA is known as a nucleic acid, or the genome of a virus. Each strand of a nucleic acid is made up of little subunits called nucleotides. If you've ever seen Tibetan prayer flags, you'll know what I mean. Each individual flag and the section of string it is attached to is known as the nucleotide. The nucleotide is also called a monomer, or more technically, a mononucleotide, where mono means one. Since the prefix poly means many, a prayer flag string that has many flags hanging off of it is called a polymer, or polynucleotide. When two polynucleotide chains meet, all of their prayer flags, the chain of nucleotides, attach to one another and form a double-stranded nucleic acid. Some of the viral nucleic acids, however, are single-stranded, or those consisting of one polynucleotide chain. If you think the definition of single-stranded viruses was too easy to be true, you're right. We're about to reach the scariest part of our horror movie. When an RNA virus is single-stranded, it must have what's known as a sense, something many B-rated Hollywood movies lack. Positive sense RNA is a strand of viral RNA that can immediately serve as a template for protein synthesis during the process of translation. Negative sense RNA is a strand of viral RNA that must first be converted into complementary positive sense strands before creating proteins. Each positive sense strand is the same sense as mRNA, which is used for protein synthesis during translation. So it shouldn't be all that dramatic when you learned that a positive sense strand can be used directly for protein synthesis. In addition, genomes that are both positive and negative sense are called ambisense, from the prefix ambi, which means both. The proteins created by the viruses of any sense are then used for their replication, reproduction, and infectivity. Keep in mind that while DNA viruses can also have a sense like their RNA brethren, this term is mainly used for single-stranded RNA viruses because DNA viruses have slightly different replication schemes. I hope all of that made sense and that I didn't leave you stranded with my explanations. Furthermore, when we talk about a single or double-stranded viral nucleic acid, we often abbreviate it as DS for double-stranded and SS for single-stranded. And when we talk about positive and negative sense, we abbreviate positive with the plus sign and negative with the negative sign. These abbreviations help us, quote, dumb down the process of writing out important stuff in our script, like the writers of cheap romantic comedies seem to often do. Finally, each of our viruses usually has an important enzyme that makes strands of polynucleotides called a polymerase, in order to help the virus produce vital proteins and help replicate its genome for the production of virus babies. The term polymerase should make sense since we define polymer as a long strand of many nucleotides or polynucleotides, and the suffix ase is used to denote an enzyme. Hence, a polymerase is an enzyme that makes polymers, or polynucleotides in our case, which are the strands of DNA or RNA in our viruses. With all of that in mind, we can begin grouping viruses by their type of specific genome using something known as the Baltimore classification system. This system is the one that puts viruses into groups, like movies are grouped into their different genres. Group 1 includes double-stranded DNA viruses. Group 2 has single-stranded DNA viruses. Group 3 contains double-stranded RNA viruses. Group 4 involves positive-sense, single-stranded RNA viruses, while Group 5 covers negative-sense, single-stranded RNA viruses. Group 6 comprises single-stranded RNA reverse transcriptase viruses, while Group 7 encompasses double-stranded DNA reverse transcriptase viruses. There are important things to keep in mind. For our purposes, DNA viruses must replicate within the nucleus, while RNA viruses replicate within the cytoplasm of the cell. In addition, the first two groups, the DNA viruses, use a DNA polymerase to help replicate its genome. The next three groups use an RNA polymerase to help replicate its genome. The final two groups of viruses have something called reverse transcriptase, or RT for short. Reverse transcriptase is a polymerase that uses an intermediate nucleic acid different from that of the original viral nucleic acid to replicate the viral genome. Basically, the reverse transcriptase helps the virus to replicate through an RNA intermediate in the case of a double-stranded DNA reverse transcriptase virus, or a DNA intermediate in the case of a single-stranded RNA reverse transcriptase virus. The most famous of these reverse transcriptase viruses is HIV, and this enzyme, the reverse transcriptase, is a frequent target of drugs used to try and stop the HIV. This is by all means just the bare bones basics of the replication strategies and steps viruses use, but I probably fried your brain enough even with that, quote, paltry amount of information. So, let's review the important points of this lesson instead. Viral nucleic acid genomes can be double-stranded. Double-stranded viruses are those that have a genome that consists of two chains of complementary nucleotides that are joined together. Some of the viral nucleic acids, however, are single-stranded, or those consisting of one polynucleotide chain. When an RNA virus is single-stranded, it must have what's known as a sense. Positive sense RNA is a strand of viral RNA that can immediately serve as a template for protein synthesis, whereas negative sense RNA is a strand of viral RNA that must first be converted into a complementary positive sense strand before creating proteins. Furthermore, each of our viruses usually has an important enzyme that makes strands of polynucleotides called a polymerase in order to help it produce vital proteins and replicate its genome for virus babies. Finally, some viruses have something called reverse transcriptase. Reverse transcriptase is a polymerase that uses an intermediate nucleic acid different from that of the original viral nucleic acid to replicate the viral genome.